Hello, my name is Santiago Sosa.
Howdy, my name is Milan Lopez, and today we're going to talk about the three different families of viruses, which we have your coronavirus, your calciviridae, and your flaviviridae. So we're going to walk you through what are the differences of each family and what similarities they have.
Okay, so first differences, we have your family Pico. First, it's small up an envelope. It's a naked virus, and it's also an icosahedral T3. It's also pseudo, and also it has a single ORF. Basically, it's just one long protein. And your VPG protein, it's attached to 5N. It's also smaller than your calcium. And the replication, it's the translation, the ribosomes bind to the IRES. Your VPG is also removed. And then basically the overall rundown of it, you got your positive RNA makes your negative RNA, and then your negative RNA is used as a template for genomes. And diseases, we have polio, your rhinovirus, which is basically a common cold, your coxsackie, and echovirus.
So then we have your calciviridae. Calciviridae is also a T3 ascohedral, which is 180 capsid proteins. So the way the calciviridae replicates in the cell is that it needs to use a VPG protein as a primer. So without that primer, you won't be able to translate your non-structural proteins and your structural proteins. So then, as you can see here, you have your RTP. It translates it into your 5' and 3', and then from there you're going to get your non-structural proteins. And that non-structural protein that will create your protein to make what makes the capsids what covers the naked virus. And its capsid, which is about 108, which you can see here, which is closer to antigenomic variation. And you see with the calciviridae, it has a 2 to 3 open reading frames, which so it has a lot more reading frames than the coronavirus, since it's a lot more bigger. So one thing about the calciviridae is that it can be grown in cell culture due to its, I believe, I forgot why, but you just can't grow it. And then you have your flaviviridae, which is also a single-stranded RNA virus, but it's also an envelovirus. So the flaviviridae has a single capsid which has a genome size of 9.5 to 12 kb, and it also known as arboviruses, which is caused by vectors such as your mosquitoes and most of your mosquitoes, because there's zoonotic diseases that affect you and how they translate it. So the way the flaviviridae replicates that, it has a 5' methyl G cap, which starts at with the translation and then goes with the open reason. But some flaviviridae viruses have an IRS. So the way the flavivirus attaches, so they have to attach with the whole cell, and the envelope will fuse, and it will be internalized by those receptors and by macrophages, and which will be through a process called endocytosis, which then translated into the ER, and then some processes are systolic. So as you can see here, you have the 5' messenger protein, and then it gets cleaved off by the whole cell proteins, and then you have your structural protein at the 5' and then your non-structural protein at the 3'. And then so fire also have an extrinsic incubation period. So meaning that it's how when the female mosquito attaches and how infectious can it be at that stage. I believe that's what extrinsic incubation is. And some example of fire is your dengue, your Zika virus, your yellow fever, your hepatitis C, and your West Nile virus. So one fact about dengue, it has 4 serotypes, which is your DHF and your DSF, which is DHF, dengue hemorrhage fever, and then you have your dengue shock syndrome. When it comes to dengue hemorrhage fever, the damage fever affects the platelets in the cell, so that's causing you to have a deadly hemorrhage and a fever. While the dengue shock syndrome affects your brother brain. And then so he's gonna talk about this. Santiago will talk about the similarities.
So similarities, we kept it nice and simple. So the first one, they all replicate in cytoplasm. Pretty cool. Next, they're all positive single-stranded RNA viruses. And last but not least, they're all icosahedrals.